Hey guys, welcome back to SciSci, and in this final lesson of cellular respiration, we're going to be talking about fermentation. Now, before we go into fermentation, I just want to review all of the steps of cellular respiration because some of these steps will actually be seen in fermentation. So let's go through this. You have three main steps. You have glycolysis, you have the Krebs or citric acid cycle, and you have oxidative phosphorylation. And we've gone over all three of these before, but just as a review, glycolysis, you have the breakdown of glucose, and you finally get pyruvate. So you get pyruvate, NADH, and ATP. Now, this is all outside of the outside of the mitochondria within the cytoplasm, and there's a small step which generates 2 NADH and produces acetyl-CoA. So it turns the pyruvate into acetyl-CoA, which is used in the Krebs cycle to produce NADH and FADH2. Now, this Krebs cycle also produces 2 ATP, and the specific numbers for NADH and FADH2 are 6 NADH and 2 FADH2. But we kind of noticed that glycolysis and the Krebs cycle don't produce too much ATP. And that's why this NADH and FADH2 are incredibly important. They've been accumulating over time in both glycolysis, in the Krebs cycle, and now we'll see them being utilized. And that is this final step of oxidative phosphorylation. This occurs in your inner membrane between the matrix and the intermembrane space. And as you can see here, it's made up of two parts. You have your electron transport chain, which is made up of these 4 complexes, and then you also have ATP synthase. So we've labeled these 4 complexes, and you can see ATP synthase is right over here. And as you can see, when NADH which is an electron carrier, donates its electron, and as it goes through the electron transport chain, we have a production of a gradient, and this gradient is used and stores potential energy that is finally used by ATP synthase to produce ATP. And this is the cellular respiration process. This is what occurs both outside and within the mitochondria. But this process requires the mitochondria. And that's what we're going to be talking about today. How do prokaryotes, which don't have mitochondria, produce energy? Now, just as a review, you have 2 classes. You have prokaryotes and you have eukaryotes. Now, we are eukaryotes. We have a nucleus within our cell, and we also have membrane-bound organelles. And one of these membrane-bound organelles are the mitochondria. Now, if you don't remember some of this information, you can look back at our tour of the cell and learn more about our membrane-bound organelles. But without mitochondria, how do we produce energy? Well, it's this process called fermentation. As you can see here in our diagram of fermentation, we're still using glycolysis because this occurs in the cytoplasm and therefore doesn't require the mitochondria. Your glucose molecule is still being turned into pyruvate and you still get 2 ATP. But this next step, which still occurs in the cytoplasm, is the specific part of fermentation, where you release CO2 and your pyruvate turns into acetaldehyde. And now once this acetaldehyde turns into your final molecule of ethanol, you see that you have some type of cycle. Now, why do we have this cycle? Well, it's because NADH produced in glycolysis, when your NAD+, which is used up in glycolysis, is not renewed, you don't have the ability to continue glycolysis. It's a limiting factor. Because without NAD+, glycolysis can't occur. And if we use up all of the NAD+ that is stored in the cell, then we can't produce the ATP through glycolysis. And here's where fermentation comes in. It uses the NADH that is produced in glycolysis from NAD+, and through the production of ethanol, it can reproduce the NAD+ molecule, and therefore glycolysis can continue. And therefore, fermentation kind of continues this cycle. Now, what is the parallel of this part, or this acetaldehyde turning into ethanol, which produces NAD+, how is this mirrored in our cellular respiration? Well, let's go and look back at our electron transport chain. If we look here, you have FADH2 producing FAD and NADH producing NAD+. And so these molecules are being revitalized. You get these molecules again, and therefore they can go back and be used in glycolysis. So now that we see the parallels and we see why fermentation is important, we can talk about the two types of fermentation. Ethanol is an alcohol, so this is called alcohol fermentation. And this is mainly used by prokaryotes. But you have another type of fermentation, and this is called lactic acid fermentation. Let's take a look at this. So in our lactic acid fermentation, you go straight from pyruvate to lactate. There is no acetaldehyde. And so this is what our muscles use. So our muscle cells, when they get fatigued and we lose oxygen and can't the Krebs cycle as well as oxidative phosphorylation, we rely on the lactic acid fermentation. And so in this process, you can see that you still get NAD+ being produced, and therefore your glycolysis can still continue. Now, this is a lower amount of ATP. You don't get the roughly 32 ATP molecules that you get out of cellular respiration, but you still get some. And so that's why I said it's used when your muscles fatigue. Now, another example of this is in yeast. You have lactic acid fermentation. And so when you're cooking bread, you actually have lactic acid fermentation being used by your yeast cells. And so now we've seen these two types of fermentation. We've seen both alcohol fermentation as well as lactic acid fermentation. And in this lesson, we've also reviewed cellular respiration. So this kind of wraps up how the cell produces energy from its food. Well, thank you for listening, and come back next time to learn about the cell cycle.